Yes, we're recording.
Okay, great. Thank you. Welcome, everybody. Thank you for coming. It's a great pleasure to have here Dr. Iain Couzin from Princeton University, who will talk to us about some of the research he's been doing actually at his lab and also in collaboration with many different labs around the world. In fact, he's on sabbatical right now at the Weizmann Institute in Israel. Dr. Couzin is a professor in the Department of Ecology and Evolutionary Biology at Princeton University, where he has the Couzin Lab, and he was a Royal Society University Research Fellow in the Department of Zoology, University of Oxford, and Junior Research Fellow in the Sciences at Balliol College at Oxford. His work aims to reveal the fundamental principles that underlie evolved collective behavior, and consequently his research includes the study of a wide range of biological systems, from brain tumor to insect swarm, fish schools, and even the big animals, the human crowds. In recognition of his research, he was a recipient of several awards, including the Searle Scholar Award in 2008, the Mohammed Dali Award in 2009, the Popular Science Magazine Brilliance 10 Award, the Pop Tech Science and Public Leadership in 2011, and last but not least, the National Geographic Emerging Explorer Award in 2012. In fact, full disclosure here, I had him actually on my list of potential guests for many years, but the one that, for many months, this year that is, and the one thing that sort of prompted me into taking definite action was I heard actually his interview on BBC. If you're a BBC fan, you would have heard his program on the exchanges for frontiers.
Exchanges at the frontier.
The frontier, yeah. Beautiful, beautiful program, and I was very impressed that obviously, and subsequently followed up on it. And it's a very great pleasure for me to give to you, Dr. Couzin. The floor is yours.
Thank you very much. It's great to be here. So how many folks in this room are biologists? Okay, so you're going to have to ask me questions as we go. So if there's something you don't understand, don't feel embarrassed about it. Don't feel you can't ask. Okay, even if we can't get to the end of the talk, I don't care. I'd rather you understand the material. Okay, so I've always been fascinated by collective behavior, the types of patterns you see here. Unfortunately, this is terribly washed out, but these are schooling fish on a coral reef, and now you can see European starlings. And we still only have a very rudimentary understanding, both the mechanism as to how, and also why unrelated organisms such as these coordinate their behavior in this way. And in nature in general, collective behavior is not only all around us, it's also within us. Your body is a collective of cells. If you cut yourself, your cells will get into collective action to heal that wound quickly before infection sets in. So we also look at cellular behavior in my lab. I'm just going to give you a quick overview of some of the projects that we work on that I don't have time to talk about today. Another beautiful example of collective behavior are social insects. For example, these ants are around 2 millimeters long, and we've put tiny little two-dimensional barcodes, actually printed by the Swiss bank, on the back of these ants that allow us, using computer vision, to track the motion of all of the individuals throughout their lives, and to look at various properties such as oscillating waves of activity that we think might have a parallel to brain waves. Another study system with the social insects, we're very interested in the architecture that organisms make. They have no blueprint, no template, and yet these simple organisms can construct these remarkable nests. This is actually a fungus-growing ant in New Jersey. And using medical imaging, we can actually get the three-dimensional structure of these nests, and also in the near future to track the motion of individuals within these structures, which of course are dynamical. I've done a lot of work over the last, almost decade, looking at locusts, a biblical plague. And so here these insects are hatching out. This is time-lapse footage of them hatching out in the lab, and they'll start marching together. And people really didn't know why or how locusts formed these large bands. And in a series of experiments, such as by making this, these are not the smartest animals in the natural world. Here's a sort of a particle accelerator for locusts, whereby we can track the motion of all of these individuals in sort of pseudo-periodic boundary conditions, allowing us to really interrogate how these organisms move and interact with each other, how local interactions scale to the types of swarms that impact humanity. And what we discovered was these apparently cooperative swarms are actually driven by cannibalism. Every insect is trying to eat those ahead and trying to avoid being eaten from those behind. So when you see coordinated behavior, be very careful. It's often not cooperative. And organisms, with the exception of some of the social insects, are typically selfish. Okay, so you have to break that assumption that we as humans often make, that when we see something coordinated, that it's indicative of cooperation. A more recent study involving Roland Case, who's the curator of mammals here at the State Museum. I'll turn the sound off. We don't need the dramatic sounds here. But Roland is a wonderful mammologist, and I think you'll see him in this video perhaps. And he's captured these baboons, and we've put GPS and accelerometers on all of the adults within a troop. No one has ever done this before. And so for the first time, we can get really remarkable data. These are twice every second we have the location of each of the baboons within the group, and this is just overlaid onto Google Maps, sped up of course, and you can really see the detailed motion and interactions. So later on in my talk I'll show you computer vision. We typically use computer vision, but for these large organisms out in natural environments, GPS and accelerometers become a wonderful means to get data. And we're also now working to apply LiDAR information to get the three-dimensional topology of these environments.
They're not ID though.
Yes, they are.
They are?
We know exactly who's whom, yes. Yes. Because each GPS has a unique identity.
What is the color?
The color here, we're using machine learning to try to automatically discriminate between different behavioral states. Because if we can reduce the dimensionality of their behavioral states, both using accelerometer data to get locomotory information, but also the GPS data, then this allows us to better tease apart who's interacting with whom. And I'll come back to that later with a more simple system, the type of approach that we're developing. The most complex organism that we work on, or arguably the most complex organism we work on, are humans. So we've been doing experiments in natural crowds in railway stations, as you see here, where we've developed software that tracks the motion of the pedestrians, but also estimates where each individual is looking. And we have actors, again, unbeknown to the people in the crowd, we have actors embedded in that crowd, subtly manipulating their behavior to look at subconscious social contagion. And so this, again, highlights the fact that we develop highly quantitative techniques to be applied with real systems, both in the laboratory and under naturalistic scenarios. And the reason we do this is, we cannot, using verbal argument alone, think through how local interactions scale to these types of complex collective behaviors. And as a biologist, what I want to do is try and get inside the head of this individual to see how and why it behaves. Now, when I started this research, there really was no available experimental data, not just a dearth of data, there was no available data. And so we started off developing individual-based models, whereby we could speculate the types of social interactions, the local social interactions that individuals exhibit. For example, if we consider two individuals, here I am showing you pictures of fish, but you can think much more broadly than that, of course. Existing at certain position C, centroid of individual I at time t, and it has a velocity. And what it's going to do is it's going to adjust its velocity, the direction, and perhaps also the magnitude, in response to other individuals. In this case, just a single other individual, but of course we can simulate larger groups. If individuals come too close together, we know they tend to avoid collisions. This can even be fatal for birds in flight. So this is just representing that turning away from collisions if you get too close to your neighbors, to maintain a personal space. If individuals get too isolated, however, this is dangerous, and so they exhibit a tendency to be attracted towards other individuals, to keep coherence, and also potentially to align their direction of travel, to align their velocities with the motion of near neighbors. And simulating these types of rules allow us to scale up from those 2 individuals up to arbitrarily many individuals. All of our simulations nowadays are done on GPUs to use massively parallel computation. Now, while it's possible with these models to create, on the left here you see the simulation, and on the right you see a video that I took from YouTube, you can see that we can capture the types of properties, such as this rotating state, that are exhibited by real organisms. But this is relatively unsatisfactory to say, oh, our model creates patterns that look like reality. But nevertheless, this is where we had to start. And so the model has a local zone of repulsion, and this outer zone of attraction, and an intermediate zone in which individuals tend to align. And what we found is even if these zones stay exactly identical, completely the same, and we just adjust speed of individuals, we can see 3 different sort of fundamental classes of behavior. Okay, so just like in physical systems, there are lots of intermolecular potentials, yet we still have only fundamental states of matter, liquid, solid, gas, and plasma, unless you really play around with the physics. We similarly predict that we have a swarm-like state, which is disordered, and then 2 locally ordered states that are globally different. This parallel group state, what you typically think of as a fish school or a bird flock, and this rotating state. And some of the properties that we're very interested in are the types of remarkable responsiveness that we see in nature. How these groups can maintain cohesion, but almost seem to behave as one, as a collective mind. Yeah.
So in your models, when you're looking at the stable states,
Yeah.
do they only depend on the parameters of the model, or do they also depend on the initial state? Or
They depend on both, and I'll get back to that question. It's a very good question, and I'll get back to that in just a second. And I'm not going to show you the analysis of the model. I'm going to show you analysis of real data, but the model makes exactly the same predictions. So both are true. Okay, so here I just added a predator and one reasonable rule, which is to move away from the predator. And previously, biologists thought that there must be a whole range of suite of different rule sets that individuals use to create vacuoles or fountain effects or dumbbell effects. Lots of terminology came in to describe these different properties. And what we found was we can explain all of these different patterns. Here you can see the dumbbell type shape that's commonly seen, just by changing the angle at which the predator attacks the group. And so really what I've become very interested in is, even though these organisms are unrelated to each other, can we think of social interactions as integrating multiple minds, allowing for forms of collective intelligence and collective cognition that do not exist at the level of the individual? So can we think of collective cognition in animals? And so in 2008, when I set up a lab in Princeton, I really had the first opportunity to try to move beyond just simulating these groups. And so I wanted to develop a system that was dynamic, had these rich dynamics, but that we could quantify the interactions. And so these are golden shiners, which we can tag with little fluorescent fish tags to know who is whom. And also, conveniently, most people in my lab are physicists. And you should have seen us down at the lake trying to catch real fish in the wild. It was a disaster. And conveniently, these fish are bred in the billions, I'm sure in this state included, for the live bait industry. For $80, you can get 1,000 golden shiners delivered to your lab. And the very first day we put them in, I'll see if I start this video again. This is a 7-foot by 4-foot tank. And you can see these are highly coherent, cohesive mobile groups. And they love shallow water. Even if you give them very deep water, they swim near the surface. And here you see them undergoing a phase-like transition between the dynamic parallel state and this rotating motion. And then with Haishan Wu, a postdoc in my group, over the last 5 years, he has developed remarkable imaging software that allows us to track the motion of all of the individuals in groups of up to around 1,000 individuals. And these are untagged fish that look to us to be identical. And yet we can actually apply a variety of techniques that allow us not to lose these individuals through occlusion. This, of course, is just showing you a central, a focal individual. I'm not going to go through the details of the tracking algorithm, but lots of the standard approaches used in computer vision do not work here. And so it turned out that the solution turned out to be a combination of novel algorithms that allow us not just to get the positions of all of the individuals, but also we map the body posture. And that turns out to be important. These organisms have got deformable bodies. And also, even though their behavior looks very high-dimensional, if you look at the low-dimensional shape space of these bodies, you find that they actually exist in this sort of low-dimensional manifold.
I was wondering if their motion patterns are also dependent on time.
What do you mean by time?
Time of the day, like...
Or time of the day. Yes, absolutely. So these fish, as you'll see, are almost exclusively visual. And we can actually dim the lights down where we can still see the fish, but they can no longer see each other. So at night, they completely disband. And again, they live in large, shallow lakes. They completely disband at night and then reform schools the next day, so not with the same individuals the next day. So light's the most important thing. Temperature probably has less of an effect. So we can then really consider these social organizations as a dynamical system, which is a common way of thinking within the engineering community, but surprisingly not a common way of thinking about dynamical processes in collective animal behavior. And so if we analyze these data, we can again think about trying to capture the complexity of that collective behavior into a low-dimensional space. So here we're plotting the rotation, which is a little bit like the angular momentum of the system, versus the polarization. In this case, a group of 70 fish for a very long period of time. In the paper, we did between 200 and 300 fish. But the point I want to make here is that the system, the dynamical system, exists in these three fundamental states of this low rotation, low polarization, or the swarm-like state, the high rotation, high angular momentum, and low global polarization. Remember, the local polarization is very high. It's the global polarization that's very low. And finally, this state that's both locally and globally polarized. And in the paper, we've also tracked how they move through this space and whatnot. But one important point that relates directly to the question before is, how are these different collective behaviors emerging? Are individuals changing their personal behavior, their individual behavior, or does there exist a metastability or bistability in the dynamical system itself? Well, the answer turns out to be both. So the transition between this locally disordered state and these two locally ordered states is driven by a change of behavior, specifically a change of speed. Just simply by speeding up, the group transitions from this state to either of these states. The transition to these states tends to occur away from the edge, which is fortunate, and is driven by ergodic fluctuations that can percolate through the group. And so this is then a group size dependent. It becomes less likely for them to change states as the group size gets larger. And so the transition between here and here is driven by a metastability. So it's not driven by a change of behavior. And if you look at the local environment from the perspective of the fish, the local polarization or the local packing fraction, for example, whether you're in a group of 30 or a group of 300, locally your environment is identical. Which is quite interesting because in models, typically when you increase the number of individuals, they become more densely packed. The real system is in what's called an H-stable regime, where density is independent. It's regulated by the organisms, and it's independent of the system size. And I'll come back to why that might be important a bit later on. So despite the fact that this model was very simplistic, it did capture those basic properties, those basic fundamental properties.
You said something for metastability?
Metastability is like a bistable regime where the system can be either in that translational motion or in the rotating motion for identical individual behaviors. And it's external fluctuations or just noise like sensory noise or turbulence or eddies that causes the system probabilistically to go from one attractor to the other attractor. So there are two attractors for identical individual level behaviors, which has an impact on how selection could operate, for example. It can't select between those two different states. Okay, so in my research, And also in the field of collective behavior, there's been an explosion. When I first went to conferences, there were 10 or 20 of us. Now there's hundreds of people, mostly physicists and engineers, coming into this field because they're excited by these types of collective dynamics and the types of collective properties. But all of these models that have been developed are what are called self-propelled particle models, where individuals interact with social forces. Now, I think the time has come for us to move beyond this dominant paradigm. It assumes that individuals average pairwise interactions, the superposition of pairwise interactions, which makes sense for physical systems, but doesn't necessarily make sense for biological systems, where there is inherent anisotropy, and also, whether I, interact strongly with you, you don't necessarily want to interact strongly with me. Organisms are not particles. They are probabilistic decision-making entities. I think we really need to put the biology back into this type of understanding. The decisions are not based on forces, but explicitly on integrating sensory information. And we've even shown in 2011 that this assumption of superposition of pairwise interactions is incorrect. At least third-order interactions are essential to understand the structure and dynamics of these groups. So what assumptions are made about the network of interactions within animal groups? Well, almost all work makes one of three basic assumptions. The previous model that I just showed you that I developed assumed that individuals interact with others within some sort of range, within some sort of distance, so-called metric interactions. Other work, based on work on starlings, has suggested that rather individuals interact with a fixed number of neighbors regardless of how far away they are, so-called topological interactions. Other researchers have argued that individuals may interact with what is effectively the first Voronoi shell, the first ring of neighbors using a Voronoi tessellation. But an important thing to note is all of these are chosen for mathematical and computational convenience. And I'm not talking about the computation of the retina. I'm talking about the computation of the graduate student, the postdoc, or the PI. So it really isn't thinking about the sensory basis of behavior at all. So what is the actual structure of interaction networks within these animal groups? Now, if we think of a comparable situation, if we consider the study of individual behavior, how individual behavior is generated, it's unthinkable that you don't study the network of neuronal interactions. Because, of course, behavior arises from the collective activity of neuronal networks. And establishing maps of neuronal connectivity has always been central since the very inception of neuroscience. However, the comparable issue of establishing the structure and nature of interactions among organisms that then gives rise to the dynamical properties of the collective have been completely neglected. There is no information, again, not even an attempt at trying to quantify what these networks actually are. And of course, this is because there are some core challenges here. The pathways of communication among organisms have no physical form. So even although in neuroscience you can't just look at the physical properties, you need to look at functional connectivity, at least there's something there to measure. Here, there's no physical form at all. And researchers, I mean, this is a really important point, researchers in the field of collective behavior are not explicitly taking into account sensory information. And so with a computer scientist, a graduate student in my lab, Colin Toomey, we've been extending this imaging software that allows us to track individuals to allow us also to automatically reconstruct where the eyes are, because it's known geometrical relationship with respect to the body and the head in particular. And using GPU raycasting technology, we can now very rapidly reconstruct the visual field, a planar representation of the visual field. In this particular image, we're actually subsampling those rays so you can see the overlap of visual fields among individuals as bright areas. As you can see, there's a lot of complexity in the pure geometry of sensing within these animal collectives. And I'll zoom in a little further so you can see a bit more detail there. And so for any individual within the group, we can then reconstruct automatically this planar representation of what it sees out of the left and the right eye respectively. So we can see what the animal sees, or a better representation of what the animal sees. Our software also works in 3D, and we're just getting that up and running at the moment to get 3D representations, because as of last week, we now have the photoreceptor density and type distribution mapped in 3D for the retina of both zebrafish and now our golden shiners. And you can also see us mapping the body posture of the individuals. Again, we want to relate this high-dimensional sensory input to the low-dimensional motor output exhibited by these animals. And just to give you a sense of the types of complexity we face here, and we haven't solved this challenge. I put this slide in a couple of minutes before my talk because I know there might be people in this audience who might have a better way of solving this than we do. But this is just representing what's seen at the left eye and the right eye, with dark regions being close objects and light regions being dark objects. And it's very elaborate because we deal not only with the dynamics of the motion of others, but self-generated motion. How do organisms make sense of these time-varying visual fields to translate that into low-dimensional motor control? And we know that there's only around 15 neurons in each side of the hindbrain that translates this into motion. Okay, so there's inherently dimensionality reduction occurring within the neural system. And so we think this could be a nice model for asking these types of questions. Okay, so to try to get at some of this, we've performed some experiments. These are real fish. Almost everything I'm going to show is real organisms, where we've trained some individuals to associate a green laser stimulus with a reward, and that will appear somewhere within this tank. And fish naturally do not have any response to this laser. And so in control experiments with no trained individuals, no one responds. And the red individuals, we know from their little color tags, have had this prior experience. And what you're seeing then is just a very simple automatic behavioral estimator that tells us when the fish has transitioned from one behavioral state to the other. So you can see this discrete wave propagating across the group. Now this wave looks very clean in this particular example. But if you look at other examples, it goes from one side of the group to the other, sometimes from the back to the front, and so on. But using these transitions, we can then directly test using Bayesian model fitting procedures between these different speculated interaction types, these abstract representations, and then this dynamic visual representation of the network. And without going into too much detail, you can see we do not have support for the previous metric and topological interactions. This is a nonlinear axis. There's much, much greater support for visual transmission in this network, okay, which makes sense. And it's kind of remarkable that this was published at the end of last year and no one had done this before. No one had actually moved beyond this paradigm of thinking about these simple interaction network topologies. We can then ask, well, what's different about this visual network? Well, it turns out, regardless of how it's parameterized, and you can see the best fits here, here, and here, you can see the metric and topological networks have different global network characteristics to the visual network, profoundly different. Notably, the visual network has a much, much lower transitivity. So what's transitivity? It's the probability that a neighbor of mine is also a neighbor of yours. So it's these connected triangles in groups. And you may think that, okay, if we have a transitive relationship, if I'm connected to you, you're connected to you, and you're connected to me, for us to message pass, we only need one hop between all of us. If we cut one of those connections, thereby decreasing the transitivity, you may expect it to require more hops to get information from one arbitrary point on the network to another. But we can measure the network efficiency, which is related to the mean path length. And if anything, the visual networks are slightly more efficient. So they're not paying a cost in terms of message passing. And the low transitivity also means that the correlation of information with neighbors is much lower. So new information can flow more readily through the network. But it's not sufficient to determine whether individuals are simply visible or not. This is a proof of concept. We only assume that individuals may be interacting if they subtend greater than a certain threshold area on the retina. Very, very simplistic. Okay, we actually, to do a really good job at this, I mean, that obviously, as I showed you, was a much better, even though it was abstract, a much better approach than previous approaches. But to do a proper job, we also need to establish the visual features the organisms actually use and the functional mapping between the salient visual cues and the motor response of the organisms. So to try to get at this question, this is all unpublished, by the way. To try to get at this question, we've been looking at what we call behavioral cascades or behavioral epidemics within these groups. And you saw the footage before of these waves propagating across the group. Well, in the fish in the lab, these are golden shiners in groups of 150. There is a reflex reaction that fish exhibit called a C-start. Okay, so if they're startled, and this can be triggered by sensory, like physical sensation or visual sensation, or auditory sensation, they will go into what's called a C shape, either that way or that way, and then burst off. It is a reflex driven by two cells called the Meissner cells. These cells tend to be close to threshold because it's very dangerous in the environments in which they live. So if we wait long enough, one of the fish will probabilistically misfire and go into the C-start and trigger an avalanche, a behavioral avalanche, as you see here. If we look at the distribution of these cascades, they have a parallel-like distribution. And all I want you to take from this is that this is a heavy-tailed distribution. So most of the time, this behavior doesn't transmit, or doesn't transmit very far. But sometimes it leads to a large epidemic. Okay?
You say those are driven by individual cells that you've located?
We haven't located them. They were located in the 40s, I believe, in Germany. But yeah, there's two giant cells, two large cells that deal with... They integrate all sorts of other stuff, by the way. But the very first response is driven by these Meissner cells. And there's non-Meissner cell fast response as well. But the initial response there is a pure reflex. Just like hitting your knee with a hammer causes it to jerk up. Yeah, so it's a classic example of a reflex behavior in biology. And the reason it's two large cells, one on each side of the body, with lateral inhibition, is that it needs to be fast. So can we determine the complex interaction network within these real groups? And really what we're interested in here is, can we use then the network properties to predict the degree to which each individual is socially influential and susceptible to social influence from others? And can we predict the size of a behavioral epidemic at the point at which the first individual responds? Remember, most of the time it doesn't lead to much, but sometimes it could be a huge cascade. Can we predict before it even happens whether this is going to result in an epidemic? So we know they respond to visual neighbors, so we won't go through that again. But what we've been able to do is actually work out what visual cues they actually pay attention to, and what the functional mapping is between these visual cues and behavioral decision-making. Now, it could have been a whole ton of different things, such as the speed of individuals, the angular position on the retina. In the literature, it's been suggested it's probably lume, which is the rate of change on the retina. Okay, so we could test among all these absolute features, but also it could be comparative features, such as the relative metric distance, the relative distance between individuals, or the relative angular area subtended on the retina, or some combination of these. Now, what we've done in this case is use two different machine learning techniques and a supercomputer to look and test between all of these different combinations for the first responder. Okay, so what we're going to do is we're going to look at who the initiator was and who the first responder is, and look to see what visual cues correlate with that individual alone. Okay? Because we don't know how they integrate beyond that, and here the causality can be readily determined. And what we find is that they respond to a logarithmic encoding of distance to visual neighbors, which makes sense because the visual system neurons encode logarithmically to increase dynamic range. And they map this with a linear addition to a comparison-based mechanism, which is specifically the ranked angular area subtended on the retina. So it doesn't matter whether individuals are close or individuals are far away. You're really ranking individuals. So if someone is subtending a large area on your retina and does not respond, that inhibits you from responding to another individual. Okay? So in human neuroscience, this very similar function has been found in how people assess how much they earn. Because there's no absolute sense of how much you earn. You compare it to peers. And so you use this kind of ranking mechanism. And what's important to note is these are both simple, fast, and bottom-up neural encoding. Surprising to me is that they do not respond to derivatives, such as accelerations, rate of approach, or rate of turning.
So is there any reason to
to believe why, from an evolutionary point of view, there might be these functionalities might have evolved. I mean, like, it looks very beautiful to the outsider to look at how, you know, a swarm of fish, how they behave. But why would this have happened?
Yeah, so there's a paper that we had in Science in 2012 that I haven't had time to talk about today, where we allow real predators to hunt and exert selection pressure on a virtual prey population. And that virtual prey population evolved coordinated collective movement. And that was related to sensory constraints on the part of the predator. That's one core reason. Another key reason, and I think I will include, I have a slide of that here, is we can, using sonar imaging, in Louisiana, we were able to look at how predators hunt prey and show how increasing group size decreases the ability for predators to capture prey. And this is driven through a scale-free correlation in terms of the information processing capabilities, the collective information processing capabilities, whereby as groups get larger, these waves of transmission are 15 times faster than the maximum swim speed of the predator. And so it's very hard for the predator to handle these big groups. Also, there are other advantages that I will discuss explicitly today regarding collective sensing and collective decision-making, which will be the second part of the talk. And so, of course, this also allows for negative feedback via that ranking of the angular area. So predicting avalanche sizes, only knowing the initiator, is non-trivial. The behavior of the initiator is not predictive. Its speed, its acceleration, its turning rate. The fish are binarizing it. You have either responded or you haven't responded. There isn't such a thing as degree of response. Once you cross that threshold, they really are, in their brains, they really are binarizing this. And properties such as the local alignment are not predictive. And we've also shown in another study that fish have got no idea whether another fish is aligned like this or aligned like this. But what if we reconstruct the predicted interaction network based on this functional mapping? If we actually map so that the edges are now going to correspond to the mapped probability that you will respond if an individual around you changes behavior. Well, you can now see for the very first time the dynamic time-varying topology of this predictive interaction network in the group, where, as I said, the edge thickness corresponds to your probability of responding, of changing your behavior in response to others. Now, this is actually a directed network, because we have to separate the degree to which you're socially influential from the degree to which you're susceptible to social influence. But you can't see that in this particular representation. It's a weighted, directed, time-varying network. So what we're going to do is freeze time at the moment that the initial individual is sort of infected or changes behavior. Can we predict the cascade size based upon the network properties at that moment in time? So any individual in here could change behavior. And we're going to then predict whether that's going to lead to a cascade or not. And we find that looking at the local weighted clustering coefficient of the individual, of the initiator, provides a very good predictive measure of whether that individual is going to be socially influential. It also predicts who's going to be susceptible to social influence. And so this might be quite surprising for anyone that knows about epidemiology, because in epidemiology, it's always considered that if you have a high clustering coefficient, you are unlikely to propagate disease widely. And so in epidemiology, and that's really been taken into animal behavior, that if you have a high degree, a high number of neighbors, and a low clustering coefficient, you're the super spreader. We find exactly the opposite. Individuals with a low degree and a high clustering coefficient will maximally propagate their behaviors to others. And so, as I said, this is work. So really, it's you know, these individuals, nodes with the high weighted clustering coefficient, that have more influence and are more susceptible. And so we can then, at any point in time, we can then map for each individual how influential we think they're going to be. And of course, this can vary in time.
So the correlation between the clustering coefficient and influence,
Yeah.
can that be considered as evidence for three-way interactions are more important?
We don't think so, because the assumptions we're making here are simply pairwise. Now, the three-way interactions might give us a better view or understanding, but it's really hard with the amount of data we have. We've got 150 events, something in that order. It's really hard to get at that higher order interactions from those data.
And clustering coefficient is computed using undirected graph?
No, it's computed as a weighted directed clustering coefficient from a paper in Physica E, I believe. Yeah, it's been established in the network literature. I should actually remember to put that citation up because I've only talked about this a few times, and I used to have it in here. It's probably in the slides that I haven't included.
It looks like you're minimizing your analysis to two-dimension here, although in nature it's three-dimensional. Do you think it's affecting?
Here these fish are two-dimensional, so they will tend to swim almost crazy two-dimensional, but almost exclusively two-dimensional. If you give them a deep tank, they will always be within the top 50 centimeters and mostly within the top 20 centimeters. And so the water here is actually about this deep, so there is some layering, which we haven't captured here, and yet still the visual network is a much, much better representation. And as you can see, we can actually predict things with this network. So it's not perfect. It's quasi 2D. But we're moving into the third dimension soon. But we chose this species because it really schools in 2D. It's close enough. Okay, so we can find that individuals near the edge of the group have the higher clustering coefficient. And by the way, the density is consistent right across the group, as I mentioned before. Individuals near the front of the group are also most influential. And so we can control for correlated quantities such as local density, out degree, distance from the group boundary. And it really turns out so far, we think, to be the clustering coefficient specifically. So using a variety of machine learning and statistical techniques such as L1 regularization and each independent variable's unique contribution, we still keep finding evidence that it really is the network structure here that is the important property rather than some correlate with the network structure. Okay, so this is sort of work in progress. And one sort of question really suggests that the interconnectedness of a node's neighbors is essential. So why do we have this difference to sort of neuroscience type understandings? Well, a lot of the work that people have done in terms of simulated cascades on networks have involved a simple contagion or a complex contagion, which is just once a certain number of individuals are infected or who have changed, that increases dramatically your probability of being infected or changing. And that's the simple and the quorum models. And as you can see, neither of them capture this relationship. If anything, in fact, is a slightly negative relationship with the clustering coefficient. It's only this fractional contagion where you're paying attention to the relative aspects of your visual scene that can explain this type of relationship. And so we're still exploring why this is the case by performing simulations whereby the dynamics are very simple, but it's run on the real complex weighted directed network, such as having k nearest neighbors or boundary k nearest neighbors or boundary neighbors triggered, and then to see how the wave propagates. And the most sort of, I guess, useful one is to look at the mutual information between cascade size distribution and whether the predator, simulated predator, is really present or absent, which is much, much greater for the properties that we see within the real fractional contagious process that we observe in animal groups, as opposed to the previous speculated means of contagion on networks. Okay, I wanted to very briefly mention that we have also gone out to the field using DIDSON, which is a new type of sonar with 1.5 centimeter spatial resolution, and tracking large number of predator attacks on real groups. And the resolution is so good we can even give a per capita risk with different group sizes. But I won't go into that. That was the study I referred to before. But I do want to talk about another property, which is collective sensing in complex environments. And really this gets at the heart of collective intelligence and comes from this chap, Sir Francis Galton. He was a cousin of Charles Darwin, an anthropologist, a tropical explorer, a geographer, an inventor, a meteorologist, protogeneticist, unfortunately, also one of the first eugenicists, a statistician, and so on. So he was a rather smart man who knew a lot about many, many things. And he was also very competitive, notoriously competitive. And in 1906, he went to a livestock fair. There was a competition to guess the weight of an ox. And of course, Sir Francis didn't know much about this, but realized, the devious sod, that almost 800 villages had already entered. And so he thinks to himself, well, the middlemost estimate expresses the voice of the people. And so he got a bit lucky. He calculated the mean as 1,197 pounds. He was 1 pound off the weight of the ox. And really, if you look at collective intelligence research, a lot of it boils down to this fundamental idea that each individual is some sort of noisy sensor that takes in information, tries to estimate the solution to a problem, and then somehow, by being an array of sensors, you can integrate these noisy estimates to come up with a better solution. Okay? And this could be done in a clever way or whatever, but fundamentally, it comes down to this Galton-type idea. So in the context of animal groups, if each individual is getting a noisy sense of some sort of complex gradient, then the conventional view, which has never been tested before, is that each individual makes these imperfect estimates. Grouping allows pooling of estimates, and in the biology literature, this is termed the many wrongs principle. Never been tested. So we set out to test this, and we created a time-varying spatio-temporal light gradient. We played around with olfaction and all sorts of other gradients, but we found light to be really good because it can't be used up. But the problem is we can't actually see the fish when they are in their preferred dark regions. So this is like a dappled stream, so to speak. This is exactly the same experiment down here, but filmed in infrared light. So this allows us to track the motion of the individuals within the group. You can see us subtracting them from the background, but because we know what we're projecting, and this has got local peaks, local optima, and also a global optima, we can then ask, are groups better able to sense these types of meaningful long-range environmental gradients? And we provided the first evidence that indeed they are able to do so, as you can see here. But when we then went to the analysis, we wanted to try to quantify over what range and how are you detecting this local gradient? And similarly, how are you responding to others within the group? And what initially really surprised us is while we had plenty of evidence that individuals were interacting socially, as you saw, we found next to no evidence that individuals were sensing, for any group size, the gradient of light. So if individuals are not utilizing the local light gradient, they're not making an estimate of the problem. They're not making these noisy estimates and then pulling them. They're not making an estimate at all. How on earth, then, are groups able to solve the problem? Well, of course, when you look at the real biology, it turns out to be something very simple and elegant. They just change their speed as a function of the local scalar measure of the light. There's no temporal integration, and there's no spatial integration. So there's no kinesis here. Oh, sorry, there's no taxis here. There's only kinesis. So they can slow down when the environment gets good. But it's a very, very poor form of regulating where you are. However, when you're in a group, this facilitates an awareness of the environment that the individuals do not have. Taxis emerges from the interactions. Because if you imagine two individuals connected with some sort of spring, and they're moving together, and then this one hits a dark region and slows down, that spring is going to get more and more taut and going to cause this individual to curve. And so that, plus the fact that, like cars on a highway, when you move more slowly, you tend to be more packed together, we could show are both the mechanisms that allow groups to collectively navigate these gradients. And it's not so much the group size that matters, it's the area that the group spans that matters. Group size is also a little bit important, but it's less important than the area. And as soon as the group spans the length scale in the environment that's meaningful, they can suddenly detect it. If they get too large for the environment, they begin to fragment as different parts of the group go in different directions. So this also provides a mechanism whereby groups can spontaneously keep growing until they match the length scale of importance in their environment. We're undertaking evolutionary simulations of this at the moment. It places minimal cognitive demands, and it turns out to be extremely robust. We looked at 3 different noise levels in that paper, and the fish were equally good regardless of noise. When strategies exist primarily at the collective level, this could have important implications for management, such as harvesting populations. So working with control theorists at Georgia Tech, we've also been able to show that this mechanism is provably optimal. And because it only requires a scalar measure and no integration, it can be implemented on very, very small robotic devices. And so we're using this now to look at collective exploration.
So optimal in what sense?
Optimal in that so this is where you'd have to look at the paper that we published with those guys, because it's got lots of lemmas and theorems. But my understanding of this is this actually maps onto a theorem in computer vision for edge tracking. And they could actually show that for a very wide range of different gradient types, it will find the optimal point within those gradients. It won't be trapped. But there's no, to my knowledge, there's no way of knowing how long it's going to take. Only simulations can give us some guidance there. But I would refer you to that paper. Yeah, that's not my area at all. Okay, in the last part, I want to talk about leadership and collective decision making. How do groups make informed, unanimous decisions? And I'm going to start with some old work before moving to some new stuff.
So I have a question about the previous models that you're making.
The previous model wasn't a model. The previous model was data.
No, about the about modeling the social behavior in the school. So can we ask questions like, given these physical constraints, what are the minimal set of rules which are required to bring about a set of group behavior? And would the reality be close to that?
We pretty much know how they perform their group behavior, actually. So that was a 2011 paper in PNAS that I sort of briefly popped up. I just didn't want to get into that. But you're right, it's a very minimal model that they're actually running. But of course, it's context dependent, you know. But you're right, yes, you can come up with a minimal model. I think we're pretty close. But so some individuals, for example, may be detecting a gradient that others are not aware of. The individuals may have learnt that there's a resource over here that others are not aware of. Maybe it's detected a predator over there the others have not detected. So I'm not going to define where it gets this internal information from. But you can imagine this is pretty common in nature. But do individuals with information have to signal to others, I know what's going on, follow me? Do others have to recognize who has and who does not have information for information to be transferred? So coming back to a simulation model for a moment, we can then simulate the social tendency, as I showed you before, but we can also capture the goal-oriented tendency that individuals may exhibit. Now, importantly, I am coloring these individuals in bright colors so we can see what information they have. There is no signaling. They start at random positions, random orientations, and there's no way of inferring the informational status. So there's no cheating going on here. And so individuals with information, we assume reconcile their social tendency with their goal-oriented tendency with a simple weighting term called omega. If omega is very low, you don't want to risk leaving the group, or you're not very hungry, and so the social tendency dominates. As omega becomes larger and larger, you're increasingly influenced by your goal-oriented behavior, perhaps even at the expense of leaving others behind. So the first thing we wanted to ask is, if we fix a preferred direction of our leaders, can one informed individual in a group of 100, or 5, or 10 guide the group? Well, if you look at one individual in the group of 100, it's not able to convey the information. The group doesn't really move anywhere. However, if we have 5 informed individuals, they begin to occupy the leading edge, and information does get transferred, albeit not very accurately. But with 10 informed individuals, suddenly and spontaneously, the information percolates very accurately and the group moves in the right direction. We can quantify the accuracy of the information transfer between 0 and 1. And as you can see, for all group sizes, as we increase the proportion of informed individuals, we increase the accuracy asymptotically. But what's really cool here is that, say we choose 85% accuracy. For a small group, a group of 10, we need around half of the individuals to have information. But as groups get larger and larger and larger, for the same level of accuracy, we now need less than 5% for a group of 200. If we start simulating cell sheets, this can go down to an infinitesimal number. So it may look as if everybody knows where they're going. But in actual fact, in reality, it may only be a small percentage.
You know, I've been thinking to myself that this idea that when you have a large group, a very small percentage of informed leaders, if you will, can collectively direct the group. This could have implications in economics because, for example, you know, imagine a capitalist system with hundreds of millions of people making decisions, and you know, the majority of them, maybe they're not very informed, and just a few of them are informed. A very small fraction, they would be really directing the entire structure.
I will come back to this issue of uninformedness later. So it's a very important point.
So does this also depend on the speed? I mean, it is not just the number of individual.
Not really. Not really. And I won't go into the details, but we've tested these ideas with the real system to indeed show that you get this increase in accuracy for the real fish. Okay, so where things become interesting, and I think it relates to what you were saying, is when there's conflict within groups. When some individuals want to go one way and some want to go another way. Can groups come to a collective consensus? Oops, sorry. I want to show you that video. And so I'm not going to go through the details. You can look at the paper for the details. But here, there's an equal number that want to get to the white. 10 want to get to the white target, 10 to the red target, and the rest are uninformed. And the group randomly selects a target. However, if I add just one more individual, you can see the local noisy nature of the interactions. Yet adding that one individual breaks the symmetry, and now the group almost always reaches the majority target. And in this paper, we sort of, we looked at the phase diagram, and it was all really beautiful. And we sort of said, isn't it amazing that they can do this, even though there are all of these uninformed individuals? Unbeknown to us, because the Nature editors only gave us 2 weeks to sort of put all of that in, unbeknown to us that it actually is because of the uninformed individuals. But if the omega value is too high, if individuals are really unwilling to give up on their opinions, then groups will tend to split. And of course, the uninformed may learn about the targets and so forth. So very recently, more recently, I've started revisiting this idea because, of course, there are conflicting interests in many types of animal groups, including human groups. And failing to achieve a consensus can be very costly. If a hiring committee doesn't agree on hiring anybody, it's detrimental to the community. And individuals may be susceptible to manipulation by a strongly opinionated or an extremist minority. And in the social sciences, it's assumed without any evidence whatsoever that those who are uninformed or have weak preferences may be particularly vulnerable to manipulation. And in human society, independently across the globe, voting has emerged. And that means it doesn't matter how strongly I believe in something, I just have the same say as you. But does this concept of fair representation make any sense for animal or cellular aggregates, which have got limited means of conveying a vote, no global overview of votes, and only relatively local interactions? But remember, they are very effective at voting through the kinematics of their motion.
So one thing I don't understand, so why is the uninformed important?
We'll come to that in a moment.
Okay, sorry.
Okay, so if we have 3 that want to go one way, say, and 2 that want to go the other way, as I told you before, the 3 will tend to dominate, to win out. Well, you may then ask yourself, why don't the minority just have a sort of arms race and become more strongly opinionated? Does that work? Well, in groups where everybody has a preference, and say the majority preference is fixed at 0.3, and I'll show that these exact values do not matter one little bit later. If the strength of the minority preference is also 0.3, the majority wins 100% of the time. But as the minority increases the strength of their omega, they become intransigent to change, and they begin to dominate, and then completely dominate the decision-making process. So 0.5 here would be half of the time the majority wins, and half of the time the minority. But that was where everybody has a preference. What about if there are groups in which there are uninformed individuals, or individuals who are perfectly well-informed, but are unbiased? They don't care what the outcome is. Well, that changes everything. So now we're going to look on the x-axis at the number of uninformed individuals in the group. The minority has a stronger preference than the majority, 0.38 to 0.3, but it's not so strong. In fact, with no uninformed individuals, the majority will still win 60% of the time. But if I put in 10 or 12 stupid individuals into the group, it pushes control back to the majority before it then flattens out due to having far too many uninformed individuals to make any decision. I can increase the minority preference strength further and further still. Now, 98% of the time, the minority will win. But if I put in 10 or 12 individuals that don't care, they push control back to the majority. i.e., they reinforce the majority view regardless of the strength of opinions. So we wanted to ask, is this a general principle? Because there are many systems where individuals can influence and be influenced by others in populations. There are technological systems, there are neural systems, and of course there are social systems. So we developed 2 models, one termed an adaptive network model and a convention model from the social sciences to investigate this. So we just looked at binary opinions, but we've gone on to show that this works with coupled oscillator models and non-binary opinions. And I'm just going to talk about the adaptive network model today, which has worked with Thilo Gross and Couzin. And so it's very, very simple, ludicrously simple. Individuals are just represented as nodes on a network with edges representing interactions. And in this case, they differ in their opinion regarding what color to be. And they have a small probability of changing each other's opinion. However, all we assume is there's a slight synergy. So if your probability of changing opinion is Q when you interact with one individual that disagrees with you, if you interact with two, it's now slightly greater than 2Q. So very uncontroversial. And so now this individual has a higher chance of changing its mind and turning green. And of course, this is trivial when you deal with 3. So we can simulate 10,000 nodes on a network. And what we find is if the majority and minority have equal strength of opinion, the majority wins. The majority view wins out. But if the minority becomes more strongly opinionated, as we saw before, they can win out. But as we change the proportion of uninformed nodes in the network, initially nothing happens. They have no effect whatsoever until a tipping point, at which point a tiny fraction of individuals that do not care what the outcome is completely dictate what the outcome is. So it completely changes the dynamic. And the nice thing about this model versus the full spatial simulation is that you can use moment expansion and moment closure to actually create an analytical closed form solution to this. And this is well beyond my abilities. This is Thilo and Couzin. But the nice thing is you can actually see the simulations as circles and the analytical model as the solid and dotted lines. You can see that there is only one stable state, the minority dominating state below a critical density of uninformed individuals. And then the appearance of what's called a saddle node bifurcation and a stable majority state. And the system flips onto that attractor. And there's also an unstable, undecided state. And the nice thing there, of course, is we can then not deal with all of the complexities of the models and look at the relative strength of minority preference versus the numerical advantage of the majority, the two things that are fighting it out in this opinion dynamics model. And you can see that the minority, if everybody has a preference, the minority can do pretty well by being intransigent. But that's with no uninformed individuals. In the presence of uninformed individuals, I'm going to now show you the region that flips from minority control over to majority. And I'll show it as white. And it's a huge area. So now the majority wins in this region of phase space, and the minority can only dominate by becoming intransigent here. So the process of being strongly opinionated dynamically just cannot work. And we make no judgment regarding the intrinsic value. I'm not saying this is a good thing or a bad thing. I'm just reporting the dynamics. But we do conjecture that it's widespread. So say you have a neural network, for example, and it needs to come to a consensus as to whether the world is moving one way or the other. One way of doing it is to create each neuron really, really well. So they're very good at encoding their direction selectivity into reporting that as a neural firing train. Okay? Well, one way of solving it. Another way is let them be noisy. And then add in completely untuned neurons to the percentage of about 10 or 12%. And that should speed up decision making and make it much more accurate. So we also have some work that shows that. But finally, we also make a testable prediction that uninformed individuals inhibit the influence of a strongly opinionated minority and return control to the numerical majority. And if we go back to the swarm model, we should expect to see a very large effect for a very small absolute number of untrained, naive individuals. And so going back to the fish, our golden shiners, we trained individuals to have a preference for, so Christos Wanu, my postdoc, did these experiments, trained them to have a preference for a blue target or a yellow target. And they have an intrinsic color-based learning bias such that those trained to yellow have a much stronger preference. And then we can put in just everybody with a preference, minority versus majority, and the minority wins because of the strength of their preference. However, if we put in 5 or 10 uninformed, untrained individuals, as we predicted, this changes the dynamics and flips control back to the majority. So uninformed individuals inhibit extremism and promote democratic outcomes in animal groups. I'd like to thank you all very much for listening and asking such cool questions.
Yeah, we have time for some. Actually, I should also let you know that he's also giving a public lecture next week in the math department on Tuesday?
Monday.
Monday.
4:30 p.m.
But I will talk about some of the same stuff, but I won't talk about any of the network, the networky stuff. I'll talk about locusts instead.
Just FYI, there is a public, so it's less technical.
I'll talk about that cannibalism story.
Yeah.
But maybe I'll take a question from here, because...
Yeah, the
Ask too many.
question that I have is about informing your animals when you were running real life experiments. So how did you do that?
So we used what we now know to be an inherent color-based learning bias. So, I mean, if you look at bees, for example, and you train them to purple versus, say, green, they learn really quickly to purple. They've evolved to learn very quickly to purple. So for exactly the same training regime, those trained to a purple stimulus will just learn more quickly and more effectively and have a stronger preference. So the same goes with fish. Perhaps because blue is not a color you really see often in the underwater environment, they learnt to associate the yellow cue much more readily. So that's what we used. You could, for example, change the amount of training. You could, for example, change their motivational state. We didn't. We kept those constant and used this innate color-based learning bias based on one paper on zebrafish. This is also a cyprinid. So we thought that was the cleanest way to get at that. But we wouldn't expect the results to change with other types of training regimes.
So can you apply that similar training to locusts, for example, or other?
Yeah, I'm sure you could. I mean, we did try, back in the day. As I said, I'll talk about locusts on Monday. We did try to look at things like social influence in locusts before, but locusts are a terrible model for these types of questions because of the cannibalism side of things. But certainly for other insects, it would be cool to think about this. Yeah. Yeah, I mean, cockroaches, for example, people have looked at, Jean-Louis Deneuberg has looked at consensus decision-making. It occurs over a much longer time scale as individuals move between shelters and so on. But it would be possible, I think, to do experiments like that. We are trying to train ants to have different strengths of preferences and look at collective decision-making in that system.
So in your results with the minority and majority, is there a phase transition in the percentage of the minority?
Great question. So there isn't a phase transition. I think maybe we're thinking about the same thing. There is always a phase transition, but the critical proportion of uninformed individuals that you need for the phase transition to occur changes according to the parameters. So if you look at the supplementary material, we show that, but it changes kind of continuously. It's always greater than 2, and it goes up to, I think, around 20 for the spatial simulation. But yes, so basically when I'm plotting that phase diagram, we're always above that critical proportion. But I think in the analytic model, it stayed around 11 or 12%. But it's a good question. For the spatial model, certainly that number changed as the parameters changed.
You say a strongly opinionated minority has chances of winning. So
yeah.
So does it affect the preference of the majority? Like if minority repeatedly opinion.
It could do. So we're doing experiments now and models now where we look at associative learning in this context. And if, for example, you don't have any uninformed individuals and the minority dominates, then the majority may then change their opinion and learn about that reward and become effectively supporting the minority. It's a really important point, actually. And so this could really change a lot of things. Yes, that could happen.
You mentioned ants and the behavior leaf-cutting ants, where these colonies up there, and they build a bridge out across something else there, which is sort of into the unknown. It's not the path that they have taken before. How does that fit in? Any conjectures of how
we work a lot on army ants. So I think army ants are the really beautiful bridge-building ones that every day they have to go out and raid and they don't know where they're going. And so there we're looking a lot at how the types of feedback, how ants sense the density of traffic over their own bodies when they're in a bridge and dynamically can change their behaviors to allow these bridges to dissolve when they're not being used and to form where they're used. So I don't think it directly relates to some of these issues, but it definitely relates to the core issues of self-organization and local interaction scaling to complex structures. And also the geometry is really important there, the physical geometry. So we've just come back from the field where we've got 3D imaging of the bridges as well, because the way it's a tensegrity structure. It's an amazing architectural structure, and we want to try to understand that. But they're a cool system, but not directly relevant to the consensus type stuff.
while modeling, are you taking into consideration the social position of an individual, of how much it can influence others, like gender or age?
Yeah. So again, these shiners turn out to be, in a way, a really good system or a bad system, depending on your perspective. They don't have any of that type of structure or hierarchy or anything like that. The individuals are completely randomly associated within the group. Now, if you look at zebrafish or medaka or almost any, a lot of other fish, they have dominance. And so then you have to take into account the structure. And so in all of our experiments on zebrafish, which I haven't talked about today, we do take that into account. And we actually find that we're interested in what leads to an individual becoming high in that hierarchy and becoming a dominant individual, both in terms of reproduction, but also in terms of decision making. And it turns out, this is unpublished at the moment, but it turns out cumulative advantage is really important. If you just get that little edge, that informational edge over the others, this can lead to an autocatalytic process where you can informationally dominate other individuals in the population and stop them from learning about resources. Because even though they go with you, you've exploited that resource, and there's no way for the learning algorithm inside that animal's head to then work. So the more information you have, the more information you're likely to get, and the more likely you are to be dominant. So for many systems, and the baboons is another example where they have very rich dominance interactions.
All right. Okay. Thank you very much. Let's thank our speakers.